We're very pleased to have the next speaker, Dr. Kanta Subbarao, with us. Kanta is really one of the world's experts on influenza virus. She was previously the director of the WHO's Collaborating Center for Research on Influenza, located in Melbourne, Australia. Prior to that, she was chief of the emerging respiratory viruses section of the at the NIAID at the NIH, and then before that, chief of the molecular genetics section of the influenza branch at the U.S. CDC. So really, tremendous background. She's currently in Canada at the Laval University in Quebec, and a professor in the Department of Microbiology and Immunology at the University of Melbourne. We are very much looking forward to what you have to tell us about respiratory viruses.
Thank you very much, and thank you for the opportunity to speak to you today. In a tradition that I have learned over 8 years in Australia, we begin seminars by acknowledging the traditional owners of the land on which we work and live, both in Canada and in Australia, and I'd like to do that. So I'm going to talk to you in more general terms about pandemic respiratory viruses. We're coming out of a long 4 years with COVID-19, and as you've heard many people talk about H5N1 avian influenza kind of sitting on the horizon. So there's really no definition of a pandemic virus, but there are some features that they share. And these are the features that pandemic influenza viruses share. And you will see that they're actually quite similar to what happened with COVID-19. So the first is the isolation from humans of a novel virus to which the general population lacks immunity. The second is the demonstrated ability of that virus to cause disease in humans. And the third, and very importantly, is the ability of the virus to spread efficiently from person to person, expressed as sustained chains of transmission causing community-wide outbreaks. So SARS-1 and MERS met the first two criteria, not the third. SARS-2 was the first coronavirus in our lifetimes that actually was able to transmit efficiently from person to person. So I recall hearing around the 1st of February of 2020 about spread from an asymptomatic individual and also spread from person to person. And I thought, this isn't behaving like SARS, it's behaving like influenza. And H5N1 viruses have also met the first two of these definitions, but not the third. So the third is really something we need to understand. So there are many features that are shared between SARS-CoV-2 and pandemic influenza viruses. They emerge from an animal source. There is opportunity for genetic exchange, and I'll talk a little bit about that. The mechanisms are different between coronaviruses and influenza viruses. And then there's airborne spread, which I just mentioned. And the surprise with SARS coronavirus 2, first of all, it was a surprise that it emerged and caused a pandemic. We were all expecting it to be another influenza virus. But the surprise was how much genetic and antigenic change occurred. We had all sort of hoped that we would have a situation like measles, where we would have a vaccine that would last 50 years. But in fact, the virus underwent a lot of genetic and antigenic drift. And of course, that had implications for vaccine strain composition. So how influenza viruses exchange genetic material is twofold. One is by a phenomenon called reassortment. So influenza viruses have a segmented genome, so think of them like chromosomes. And when a cell or a host is co-infected by two different influenza viruses, here schematically shown in blue and red, each of the gene segments are replicated. And when new daughter virions form, they come together and bud out as progeny virions, but they could derive a gene segment from either parent. This happens all the time in nature. It happens in animal influenza viruses and human influenza viruses. It's an ongoing process. But you can see how a virus could derive a new hemagglutinin or a new neuraminidase or some other accompanying gene that then makes it novel to the human population. Coronaviruses, on the other hand, recombine in nature. And so normally each species has its own coronavirus, and we always believed that they would stay in their lane and rarely cross over, although there had been known instances of crossover. But what's happening in our world is that we are, very often through human efforts, are shrinking the world that our animal viruses are able to stay separate. And so we've got recombination between, say, a bat virus and a virus from domestic animals or from humans. And so this recombination then allows an exchange for genetic material and the potential to spread. So the first thing that has to happen when a novel virus emerges is it actually has to be able to infect the host. The first step is virus entry. It has to recognize its cognate receptor and get into the cell. So here are some examples of how different viruses enter the cell, but the key is that the receptor has to be present and the virus has to be able to attach and get in. And then it has to be able to evade innate immunity that we all have to varying degrees, and the viruses all have mechanisms or have often developed mechanisms to evade innate immunity. So in the case of influenza, the attachment protein is called a hemagglutinin. And it exists as a trimer, and here color-coded are two of the trimers, and the third trimer is behind. The receptor binding pocket is up here circled in red, and the receptor for influenza viruses are sialic acid. But there's a subtlety beyond that in that avian influenza viruses preferentially bind sialic acids with the 2,3 linked oligosaccharide chain, whereas human influenza viruses bind sialic acid with a 2,6 linked chain. So why does that matter? It matters because in humans, both types of receptors are present, but where they're located in our respiratory tract varies. So we have a lot of alpha-2,6 receptors in our upper respiratory tract, and more of the 2,3 receptors beyond the respiratory bronchioles into the lung. And so that has been proposed as a hypothesis, as an explanation for why some H5N1 infections cause very severe disease, because they bind the avian receptor, which is present in the lower respiratory tract. Now, this is not black and white. It's not all or none, because all of the cases of H5N1 that we've known, which goes up into the 800s, have all been diagnosed with a nasal swab or throat swab. So there's plenty of virus in the upper respiratory tract. It's not all or nothing, but that is one of the hypotheses about how you get more severe disease caused by avian influenza viruses. So influenza viruses come in 4 different types, A, B, C, and D, and only influenza A viruses can cause a pandemic, and it's because influenza A viruses occur in many different species. Now, normally, like coronaviruses, they stay in their lane, so only 3 of the 19 known hemagglutinin subtypes have caused sustained disease in humans, 1, 2, and 3. Pigs have had H1 and H3, horses have had H3 and H7, and you see the smattering. But almost all of the subtypes have been isolated from waterfowl and shorebirds. There are 2 recent ones that were identified in bats, and we're not yet fully sure how much of a pandemic risk they pose. But the viruses from waterfowl and shorebirds are in ecological stasis in their natural reservoir. They don't cause disease. They're shed from the GI tract in very high titers, so think about that when you're on a beach, but because they are out there. The neuraminidase is the other major surface glycoprotein of influenza, and they come in 11 different subtypes. Again, 2 most recent ones identified in bats, but the other 9 in waterfowl and shorebirds. And again, only N1 and N2 neuraminases have been isolated from disease in humans. So the natural cycle of influenza A viruses are in waterfowl and shorebirds, and they occasionally spill over into domestic birds, and this I mean chickens and turkeys. And they can then spill over into mammals, pigs, and I guess I should now draw a cow here, and humans. So that's the first step, is the receptor has to be there, the virus has to be able to bind its receptor and infect cells. Now, coronaviruses, on the other hand, we've had 4 seasonal human coronaviruses. that are the first four listed. Then we've had SARS and MERS and SARS-CoV-2 in the last, since 2000. And these are the receptors that they bind. And you can see a recurring theme of ACE2, but MERS binds dipeptidase 4. So I was in Australia when when COVID-19 began, and Australia had a very different experience with the COVID-19 pandemic than you did in the US. Australia, big island, they closed their borders. And most infectious disease folks will tell you that, oh, that'll buy you probably 2 weeks. It bought them 2 years because they closed their borders and anyone who came in had to go into mandatory quarantine for 2 weeks. The quarantine was a little leaky. We did have some illness, but by and large, SARS-CoV-19 was not a big issue in Australia. And I had worked on SARS and MERS, and I was very keen to work on COVID-19, but there was, limits on what I could do. And the head of cell biology at the Children's Hospital came to us and said, why are you working with African green monkey kidney cells? Why don't you work with human cells? We can engineer them to be anything you want. And I thought this would be a great opportunity to actually be able to work on the biology of SARS-CoV-2. So what they started with are pluripotent stem cells that they then differentiated into lung, kidney, heart, CNS, and others. And so we did a series of infections in organoids and cell models. This is the kidney, we did placenta, very early term placenta, but mainly worked on the heart and lung. And so what we saw, in fact, the upper panel are infectious virus titers, the lower panel is the corresponding genomic data. And what you can see is that the lung and cardiac cells could be very well infected with SARS-CoV-2. And on the left is the African green monkey kidney cells. So we saw very robust infection in the human stem cell-derived tissues. And this is what the lung cells look like. It's a little creepy, but you can see them beating. And as you infect these cells, on the left-hand side, you'll see them continue to beat. But on the right-hand side, they curl up and start dying, and they stop beating. So, but then by 4 days post-infection, so they get slower and slower on the infected side, and essentially we lose the cultures and they stop beating. So we then tested a bunch of antiviral drugs, and this is just one example, which is remdesivir. And on the left-hand side, you see in vero cells, and vero cells we knew already from Tim Sheehan's work from UNC, that the enzyme to phosphorylate remdesivir is not very robust in vero cells. But in the lung cells and cardiac cells, you can see how much more sensitive they are to the antiviral activity of remdesivir. In fact, in this inset, we had to go way down in the concentrations. So we did a series of studies looking at phosphoproteomics and so on. But I would argue that what I learned from this was that it is important to use relevant cells and to look at antiviral drug development in more than one system to see if the drugs work equally well in different cell types and potentially to have rational combinations. The other thing, so of course I learned from this that we should, even though virologists are very excited when we have a cell line that's infectable, because that gives us something that we can work with, it is great to actually also look at the relevant cultures. So we've now set up all the way going from nasal cells down to the alveolar epithelial cells, we've set up cultures of the human respiratory tract to use to evaluate novel viruses as they emerge. One of the arguments against all of this, of course, so you can differentiate them at an air-liquid interface and they form this multi-layer. All the cell types are present because we start with the basal cells. But the issue is that in the ALI cultures, you don't have the architecture of the lung. So what we're doing now is what are called precision-cut lung slices, where you can actually inflate the lung with low melting point agarose, cut sections, and put them into 24-well plates, and you can infect with any virus you want then. And that gives you some more of a sense of what's happening in that culture system. So when we look at human coronaviruses, thinking, well, maybe we should see how this human respiratory tract culture system works with conventional human coronaviruses, you can see with NL63, 229E, and OC43, we see varying levels of replication in cells going from the nose down to the alveolar epithelial cells. And OC43, interestingly, only infected our IPS-derived alveolar type 2 cells. And then these are precision-cut lung slices from ferret lungs, and you can see that we were able to infect them with H5N6 and H5N1 viruses. So this is, one ferret can give you enough lung slices that you can actually screen a whole lot of viruses to see which ones might actually pose more of a threat for mammalian adaptation. And just yesterday, I saw this paper in Emerging Microbes and Infection using calf lung slices to look at the bovine infection. So this is a 2344B virus, and this is an H5N1 clade virus. So you can see that in calf lung slices on the left are infectious virus titers, and on the right is the genome copies. So for many, many years, we believed that avian influenza viruses would not infect humans unless they reassorted with the previously circulating human influenza virus. That was true in the 57 and 68 pandemics, and we thought that was pretty much what would happen. But the reality is that we are now sitting here with over 800 cases of H5N1 infection directly infecting humans, and over 1,500 cases of an H7N9 virus that infected humans. So we have an ongoing series of infections going on around the world, and I've only boxed the most significant ones. And so there's no clear pattern. There is an exposure issue, because most people have had an exposure to infected poultry or infected cows, and as the case is now. And of course, there are new infections in 2024. So the first H5N1 human infection occurred in 1997. I was at the CDC at the time, and we characterized that virus. And it belonged to a clade of viruses called the Goose Guangdong lineage that had been isolated from geese in Guangdong province in 1996. And that virus reassorted and got its NA from a different source and its internal genes from a different source. And at this point, as of earlier 2024, there was very high case fatality rate. The viruses, the precursor viruses, continued to circulate in Asia. They were sporadically carried out of Asia by migratory birds with outbreaks in the Middle East, in Europe, and Africa. And in 2014, the viruses actually crossed the Atlantic and came into Canada and the United States. But at that point, culling the infected poultry actually ended the outbreak, and it went away until 2020. Now, what you cannot see in great detail on this side of the slide is how much the virus has evolved since 1996. It's gone into fourth-order clade, so there's a lot of genetic change. And the 2020 virus, which is called clade 2344B, like I said, it's a fourth-order clade, they got introduced into Europe and have caused outbreaks in poultry, and then they crossed into Canada and the United States, so you can see all these red dots. The virus has spread into South America. There's been a lot of spillover into small mammals that we believed were getting infected by eating dead birds, and they've now infected many marine mammals, so there are massive die-offs of elephant seals, so there's a real effect on wild populations. So they have a global distribution now, and it's considered a panzootic. So the big surprise in 2024 was the isolation of H5N1 viruses in cows. And the first signs were a drop in feed intake and milk output. Symptoms could range from asymptomatic to a very handful of lethal cases. But notably, the viral load in the milk was very high, so it was as high as 8 logs of virus in the milk. It's been almost exclusively in lactating cows and more in older animals. And on the right-hand side is what I uploaded from yesterday. The total outbreak in cattle so far have been in 718 confirmed cases in 15 states. And now, at this point, it's primarily California, but this outbreak has been going on since March. And I will note that because influenza A was not known to infect cattle, it took a while to make the diagnosis. So we have to always be aware that it is hard to diagnose something when you're not thinking that it's likely. What about human spillover? So there's a growing number of cases, human cases in the US. There have been a total of 58 cases in 2024, most of which are mild, and most of which have had a link to either poultry or to cattle. So there's one case in Canada that is a teenager who's very sick, and we don't know what that person's exposure was. But there are mild cases associated with the bovine outbreak. We don't know why they're mild. We don't know if it's because the route of exposure is different from what was happening in Asia with the H5N1 viruses before. And it's also possible that neuraminidase immunity is ameliorating that. We don't know. So the first part is the virus has to get in, it has to replicate and cause disease. But the second bit that is very important is understanding whether the population is susceptible and what part of the population may be protected, may be partially immune. So you heard before that in the case of the 2009 pandemic, people that were born before 1954 had exposure to a virus that was similar to what emerged in 2009. And so we had a subset of the population that had some immunity. And this is from the CDC. These are serologic data showing that this is the titer of antibody. And you could see that people that were born before 1950 had some antibody. So we also know that there may be some implications of your birth year and immune imprinting that was mentioned in the previous talks. The question is, do we have serologic assays and do we have appropriate controls? And do we have cellular immunity assays and do we know how to interpret what they're telling us? All of this becomes important for pandemic planning. So the next thing that I want to talk about is airborne transmission. And this is just to remind you how explosive a sneeze can be. So if you cast back to the COVID-19 pandemic, if you look at how airborne viruses are transmitted, they're transmitted through the air. They're also transmitted by fomites and direct contact. So there are multiple ways that the virus could transmit. And there's been a lot of attention on droplet and aerosol transmission. When I trained in medicine, we were taught that 5 microns was the magic number, that particles that were more than 5 microns in size would drop out of the air within 3 feet or 1 meter, which is why the beds were that far apart. But aerosol scientists tell me that that's complete nonsense, that aerosols are generated at about 100 microns and they rapidly dry up, and then you can have particles that are suspended in the air. So aerosols and droplets still exist, but the size of droplets that we emit, even in quiet breathing, are much larger than we were taught. Why is this important? Because the size of the particle, when you inhale it, can deposit at different parts of the respiratory tree. So particles that are large in size will stop in your nasopharynx. Particles that are smaller in size could go down into the alveoli. So there has been an effort now to bring the fields together for aerosol scientists to inform medical scientists, and there is a new publication from the WHO that's sort of trying to define this as particles that are traveled through the air, and that when you're close to somebody, very often you're exposed to the larger droplets as well as aerosols, and when you're further away, it's aerosols, but it's not black and white. It's not that you get droplets at one distance and aerosols at a different. There are many traits that are associated with airborne transmissibility in mammals that are viral characteristics. But in addition, how do we study this? So I think Peter mentioned we use a ferret model because mice don't cough or sneeze, and they don't shed a lot of virus in their respiratory secretions, but ferrets do. So ferrets cough and sneeze and get a fever. And we can separate ferrets with the mesh and look at airborne transmission. And this, you will see, comes up repeatedly when novel viruses emerge as to whether respiratory droplet transmission occurs. And we know that there are some viruses, H5 and H9, that did not transmit in ferrets, and they haven't transmitted in people. But there are two viruses, subtypes, that did transmit in ferrets, but they haven't transmitted in people. So we wondered why that was and thought, well, maybe if instead of just looking at an infected ferret and its neighbor, we should look and see whether there would be sustained transmission, so the next generation. So my prediction was that sustained transmission would fall apart with sustained transmission, but in fact it didn't. So once a ferret was infected, it could transmit. But so what I conclude from this is that the transmission in the ferret model is necessary, but it's not sufficient to predict transmissibility in humans. So when a virus doesn't transmit in ferrets, then it really doesn't transmit in people. So those are the no's. But when it does transmit in ferrets, we still have to, we can't be convinced that that is predictive of what's going to happen in people. So I'm very interested in airborne transmission, and it really is going to take a multidisciplinary effort to understand airborne transmission. We're working with epidemiologists, infection control physicians, mathematical modelers, and aerosol scientists to understand this. So what happens with severity? The first definition that I gave you didn't include severity, and the WHO was very severely criticized for not including severity in 2009. And there were countries that said, well, this wasn't even a pandemic. So there has been an effort to try to get people to understand how to monitor severity. And so the WHO has this severity assessment and a guide on how to do this. So there's a universe of viruses, coronaviruses and influenza viruses out there in different animal species. Which of those pose a threat? So the WHO has recently gone through an exercise to look at virus families and identify which of them pose the greatest risk, and the ones in red pose a high risk. And if you look at these maps of the world, this is influenza. So it is something that really is important for the whole world to consider. So how do we assess the risk of a novel virus? So the WHO has a system that they call TIPRA, the WHO tool for influenza risk assessment. The CDC has something similar. In fact, theirs was first. It was called IRAT. And what we do is we look at different elements. We look at virus characteristics. What are the receptor binding properties? What are the genomic characteristics? Is there transmission in animal models? And is the virus susceptible to antiviral drugs, the known antiviral drugs? We look at attributes in the human population, human infection, how severe the disease was, and whether there's population immunity. And then we look at virus ecology. What is the geographic distribution in animals, and have there been infections in animals? So experts choose which of these topics they're comfortable scoring, and we look at the data, score the data, and then the WHO weights those scores, and they come up with, they ask us two questions. What is the likelihood that sustained human-to-human transmission will occur with this new virus? And what is the impact on the human population if it is able to transmit? So these are two separate questions. The triggers to do a TIPRA exercise are rapid geographic spread of a novel virus, an increase in the number of animal species affected, the spread in non-human mammalian hosts, and then their virologic triggers. From looking at the sequence data, are there features that make us worry? So this is what it looks like. So on the x-axis, you see the likelihood of emergence. So you score that in that direction, but then the impact score could be quite different. So you could have a virus that is likely to emerge, but would likely have a low impact, or you could have something up in this upper right. So there's a good review of this in Lancet Microbe. So what do we do with all this information? This is NIH and WHO both agreed that once you identify virus groups, you really need to develop the basic preclinical and translational research to address these issues and then move them forward into clinical trials. What are some of the lessons from COVID-19? Surveillance. So we need to do more of this in the case of H5 in agriculture and dairy producers. Financial considerations complicate the picture. We need to protect ag and food industry workers. including compensation for quarantining and for medical care. We need inexpensive, non-invasive rapid tests. We need non-pharmaceutical interventions. And of course, key is communication, communication, communication. So One Health is the idea that the health of people and the health of animals and our environment are intertwined, and we cannot achieve one without the other. But we talk a lot about vaccines, and this is to bring it back to the whole world. How accessible are vaccines globally? If you can't see in the back, this is upper middle-income countries, high-income countries, lower middle-income countries, and down here are low-income countries in terms of access to vaccines. And in this one bit of data, high-income countries have to increase their healthcare spending by 0.8% in order to vaccinate 70% of their population. But low-income countries have to increase their healthcare spending by 56% to achieve that goal. We don't just need vaccines, although we are nowhere without them, but we also have to make them priced affordably, allocated globally, and widely deployed around the world. And this is some of the other writings that have been published about the production, allocation, affordability, and deployment. So just to close, potential pandemic threats are, their worst case scenarios are when there's no immunity, when they're airborne, when there's silent transmission, and if there are no existing vaccines or therapeutics. But fortunately, there are lots of people working on different aspects of this. So I'm going to close by acknowledging my colleagues in Australia that gave us the IPS-derived tissues. I have a big group that I work with on airborne transmission. This is my own research group, Still Smiling, and most of my funding came from Australia and now the Canada Excellence Research Chairs Program. And I've just set up a new center on biology and control of zoonotic and pandemic respiratory viruses in Quebec. So thank you for your attention.
Thank you. Questions?
Hi, Kanta. Thank you very much, Greg Martin. What is the logic on why the influenza viruses occur every year? So what is the reasoning behind that?
Very good question. We often wondered, we spent a lot of time wondering where they went in between seasons, and whether viruses were seeded from the southern hemisphere into the northern hemisphere or the other way around. So the seasonal influenza does not come from an animal host. Those are human influenza viruses that continue to circulate, and at any given time, they are somewhere in the world. So the WHO has a very, very good global influenza surveillance system where each country was encouraged to have at least one national influenza center, and some countries have more than one, and they routinely monitor what's going on. So we do have influenza circulating around the world somewhere, and that's what gets reseeded into the population.
Thank you so much for that talk. So has the mutation that allows for the replication of H5N1 in cows been characterized? And you suggested that maybe these infections to humans are mild because of prior immunity. What contribution would that mutation that allows them to infect cows be associated with milder infection?
So we don't know of any specific mutation that enabled infection of cows. What we know is the receptors are present. So we know that the alpha-2,3 linked sialic acid receptors are present in the mammary gland. So there's a lot of 2,3 receptor present. So how the first cow got infected is unknown. And we assume it was originally respiratory, but the further spread is through milking equipment, we believe. And so whether it's retrograde through the teats while they're being milked, because the milking equipment is not cleansed between cows. So it's primarily a mastitis with very limited respiratory infection, but there's a lot of virus present, and it is possible, and experimentally, the infection has been kind of replicated by doing intramammary injections. So we don't know of any specific mutation or whether it just was a chance occurrence that an avian influenza virus was present either in the feed or in the water or in the environment that infected the first cow, but we don't know.
But presumably the highly pathogenic strains have been sequenced, and so has the cows more modern strains, and there must be differences between the two strains.
There are some differences, but nothing that stands out. So it's a slightly different clade than, so what's out there in wild birds is genetically quite diverse, and one particular clade got into cows, but the other clades are out there. So the infection in BC in the teenager is from a different clade. And there was a case in Oregon on a farm where a pig got infected, and those two belong to the same clade, and they're different from the clade infecting cows.
Can I follow up on that? There have been recent reports, I think. There have been recent reports that the cows in Europe are now also infected. No?
I didn't know that.
Oh, maybe not. Okay.
No, you may know, no.
I was going to ask you how that occurred.
Probably, yeah, no, I don't. Well, the precursor viruses are in wild birds.
Okay, I think we have to recognize that, of course, WHO and the global health community has been reluctant to accept the fact that this virus is airborne. And of course, as you pointed out, we humans, I mean, we talk, sing, shout, and of course, we are creating these speech droplets. And our colleagues, and including us, we actually demonstrated that these 2 micron, 3 micron sized particles, speech droplets, of course, drying out and becoming ever smaller, and they float in the air several hours. I mean, we have using laser scattering to visualize this, right? And masks do work, and there's so we have to make sure that I think when, as a preparation for next pandemic, that I think WHO and all the people need to communicate to the public that this virus is airborne, and I think we have to make sure that the ventilation systems, all the public health measures are actually kind of take that into account. And one other thing that I think I would like to mention is I think antivirals could also be a really effective tool to contain. So I think we should also include that, because that's usually kind of these two things, airborne and antivirals, are usually dropped out in the from the conversation. So thank you.
Yeah, no, I couldn't agree with you more about the airborne part. I just never understood why masks got so politicized. So a very interesting way somebody put it is that when you're face to face with somebody, you're in their plume, and otherwise it's in the room. So what's in the room, which are the finer particles, can be cleared with ventilation, but no amount of ventilation will correct the plume. Masks are needed for that, masks and distance. So I do think we are getting closer to accepting, and I don't understand why that was so much of an issue, but I will say that the N95 or P2 masks work very well, but when they're fit tested. And there was very little fit testing at the very beginning. So I think we have learned a lot from the COVID-19 pandemic that needs to be moved into our thinking about influenza. But even right after COVID-19, I had people call me and say, but flu's not like this, is it? And I thought, why would you say that? So why do we have to revisit this? But true. And this medical countermeasures here is actually vaccines and antivirals.
Just again, following up on the depoliticization, I can't say the word, of masking. So one of the things that had happened in that first year was the widespread dissemination of a study that claimed to show that masks were ineffective. And so are there strategies that can be used to provide
More data.
both what is wrong with that study and what other evidence there is that could be transmitted to friends, neighbors, family members who have seen that one study and said, I'm done with a mask.
Yeah, yeah, no, I think that you're right. I think we do have to make sure that the information or newer information is communicated and communicated well. I think what we've learned is people stay in their echo chamber and aren't really open to hearing new stuff. So we have to find a way to do that.
I have a very simple question, and one that's been bothering me as I keep reading about H5N1 in cows and in other mammals, and the few instances we have in humans. How good and how extensive are our tests? How much do we have? Who is controlling that? It can't be just in the United States. We live in a globe. These viruses know no borders. What about the testing? Who's controlling that? Who's giving the funds to do it? And what is our future? You've stated a lot of numbers, and those numbers are not terrifying, but I'm terrified because I don't know all the numbers.
Yeah,
And so,
I'm sorry. You said it was a simple question, and I knew right then it was a problem. So I don't actually know who controls things, and maybe Deborah will answer that in the next presentation. But I do know that, for instance, in Canada, they're very worried about what's happening in cows in the U.S., and they're very worried about the virus crossing the border, aside from the wild birds that are disseminating it on their own as well.
Yeah.
So I think testing is insufficient. That would be an understatement.
Yes.
But who's controlling it is something I don't know. So we do need rapid, affordable testing, and we need to make it available to people that don't trust government. We have to find a way to anonymize it or in some fashion keep that information restricted. Otherwise, there will be no uptake.
Thank you.
I don't think it's come up, but I thought one of the most remarkable things about the pandemic was how there was almost no flu cases in the critical years. So in other words, masking and distancing and lockdowns
It did
worked
work.
great for flu, just not for COVID.
Yeah, you're right. So I think that many of the social and preventive measures that were put in place for COVID-19 worked for flu. Almost everybody around the world saw no flu for about 2 years. There was a little pocket in the tropical belt where there was some influenza because the WHO was quite concerned that people would stop looking for it because the same labs pivoted to being the COVID labs. And so they really pushed these national influenza centers to keep looking for flu, and we were looking for it, and it was there in pockets. So when the virus reemerged, when flu reemerged, we were very curious about what would happen to that genetic bottleneck. And the one benefit of COVID-19 is that one of the strains of flu B seems to have disappeared. The B Yamagata lineage is gone, as best as we understand it.
Kind of to follow up on that and how these strains change over time. So you talked a lot about how you get the recombination or the segmentation for the flu virus and that genetic shift, and how you were surprised, or scientists were surprised, on how SARS-CoV-2 became so shifty. So was there something, so if you go back and look at these SARS viruses kind of in their native habitat, do you not see that sort of genetic shift there? And is there some sort of genetic basis that has been uncovered about why that became? Was it just the pressure in a new population?
Yeah, that's a really good question, because usually when a virus is in its natural host, there's less pressure for it to evolve. What we do know now, looking back, because the human coronaviruses were understudied, but now we've gone back and looked at them more closely. 229E actually does undergo a lot of genetic drift and antigenic drift. And OC43 has 2 different clades that are... So there is more out there of drift than we thought. But we don't have vaccines against them, so we don't actually know how much they're evolving antigenically as well. They might be. But I think the surprise was that coronaviruses actually encode a proofreading function. So we had sort of hoped that errors would be corrected, and they're not. But we also have had a global pandemic, so the sheer volume of virus and the number of hosts infected may be why the XON was just overwhelmed and couldn't correct. Now, once the virus settles into a human seasonal pattern, we may see less of it. But I don't know that for a fact.
Can I just point out that it actually follows that nobody expected rapid antigenic change from SARS-CoV-2 because, like in the veterinary context, there's like IDV chickens, and that's infamous for the incredibly dramatic changes in the sequence composition and the genetic properties and so on. And there are such cases. So it was actually everyone who knew anything about coronaviruses, which wasn't me at the time, but I did my homework, and knew this was going to happen. Just so you know.
Thanks.
Like, the moment, like in January, February 2020, it was known that vaccines were going to fail, just so you know.
Any other questions? Okay, if not, thank you very much.